Good evening, grade 9. So hopefully you had a good weekend and you're ready to kind of get started with next week. We're going to have 2 lessons only this week. This one, lesson number 6, is for Monday before your quiz, which is on Tuesday. And then you'll have another lesson probably posted on Wednesday because our last day of school week is Thursday. Since we only have a 4-day week this week. So we're talking about evolution of populations. So instead of talking about individual organisms, we're going to look more at the population scale, which is where evolution really happens. So here's your template for today. There are 2 drawings you have to draw. So take a minute and pause the video and draw this in your notebook, please. We'll be uploading a picture of this to Schoology for today's lesson. Okay, so hopefully now you've drawn your template. Start talking about the information for today's lesson. So the first thing is gene pools. Have you ever heard of the term gene pool? And if you have, what do you think it means? Pause for a second and think about this. So a gene pool is depicted here in this drawing with all different kind of butterflies. And you'll notice that's one of the ones you have to draw, the first drawing you need to draw. And the different colored butterflies all have different letters inside them. 2 capital A's for dark brown, 2 little a's for the white, and a big A and a little a for the orangey light brown. Make sure you include those letters in your drawing. They are important. So what a gene pool actually is the total number of genes in a population, specifically among the reproducing members of the population. So that's a really important part of this here is the reproducing members and the number of genes. So remember, genes are in the DNA, and they're what codes for all these different things. One of the 4th part of natural selection is descent with modification. So we have to have DNA to be passing these traits on for that to function. Okay, so a reproducing member. What does a reproducing member mean? What does that mean? What do you guys think? Pause for a second and think about it. So a reproducing member is any individual who is of reproductive age. So they're not too young that can't reproduce, not too old that can't reproduce. They're in that population and they're able to reproduce. So it doesn't include the genes in the very young or the very old, just the reproducing members in the population. Okay, what is the gene pool, the size of the gene pool? How is that connected to variation? We know variation is very important for natural selection. So how do you think the size of a gene pool will be connected to that? Well, if we have a larger gene pool, we're going to have more variation. It means we have more different genes in that population. There's more variation, which means in the end we're more likely to have the ability to adapt to an environmental change. Okay, so some things can reduce variation, and one of those things is inbreeding. So does anybody know what inbreeding is? So inbreeding, in case you don't know, is when we have 2 related individuals, usually siblings, who will reproduce. So if you look at this little picture here of the bunnies, we have 2 bunny parents. Oops. Okay, one parent, the other parent, and they reproduce, and they have 3 babies. The variation here, and these 2 babies are going to reproduce to make this baby. That's inbreeding. These 2 babies, because they're siblings, producing their own child, that is inbreeding. Okay, so how would inbreeding affect the size of a gene pool? Well, it's definitely going to decrease variation because instead of mixing between different individuals and having this individual mate with from a totally different parentage and maybe have different alleles, we're just mixing the same alleles around. So we're going to have these traits showing up that maybe wouldn't show up otherwise. So it actually decreases variation in the population. Now, in most modern societies today, inbreeding is not acceptable, but in ancient Egypt it was very, very common. Does anybody know with their history, why was this common in ancient Egypt? Well, the big reason why is because in Egypt it was thought to be keeping the bloodlines pure. That if a pharaoh married his sister, that they would keep the bloodlines pure. But this actually caused lots of problems for health-wise. It has a huge decrease in genetic variation, of course, and then diseases and traits that are recessive and wouldn't show up normally become much more common. So today we understand this, but they didn't back in ancient Egypt. Okay, so let's talk about kind of where this genetic variation is coming from. So if we look here, start with the kind of the cell. Here's a cell, and inside the cell we have the nucleus where the DNA is, right? I think we learned with the cell before. So that DNA is organized into chromosomes, and that chromosome is made of DNA that's been kind of folded very carefully to make up into a chromosome. Okay, and these little bases here are what makes up the code that tells what it is, what to produce. So here's two different strands of DNA that have the same gene on it, coding for the same thing. Let's say in this case it's eye color. And this one, while they both code for a color of the eyes, they code for a different color, and that means they're an allele. So an allele is the different version of a gene. So this gene, this sequence in the DNA, let's say it's eye color is a good example. This one codes for blue eyes. This one codes for brown eyes. They both code for eye color, so they're the same gene, but they code for different eye colors, therefore they're different alleles. Okay, another version, here's one chromosome, another chromosome in a flower this time. It's the same chromosome, and the DNA here is organized, and this right here, this allele is for a purple flower, and this allele right here is for a white flower. So capital P is for purple, and little p is for white. It's the same letter because it's the same gene, but different alleles. So one's uppercase, one's lowercase. So you can think of the allele as the unit of genetic diversity. So all the genetic diversity we talk about and we see in populations that was necessary for natural selection and evolution to happen, that comes from the DNA. And so the alleles are the different versions of the same gene, and that's where this variation in population comes from. Okay, so let's look at how we analyze how frequently an allele shows up in a population. That's called allele frequency. So if we always keep in mind that alleles are different versions of the same gene, okay, just like different colored genes, different colored eyes in the gene for eye color. Now the allele frequency is the percentage of individuals in a population who have that particular allele. So for example, in Morocco, I know that blue eyes in Moroccans are not very common, but they're much more common in Canada. It's going to be a different allele frequency of eye color of blue eyes showing up in Canada than there would be of blue eyes showing up in Moroccan population. So the example we're going to look at here and do together is pea flowers. So there's two options. You can have white flowers, okay, and the white flowers are going to be the little w, two little w's, and the big W makes purple. Okay, so we're going to take a minute. I want you to calculate the frequency of each allele. So what is the frequency of the big W allele, and what is the frequency of the little w allele? So the first step is just to count up all the big W's. So we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13. So we have 13 big W's out of my total of how many do we have? 1, 2, 3, 4, 5, 6, 7, 9, I have 18. So 13 out of 18 means I have 72%, or 0.72 is the allele frequency of the big W. The total number of alleles here, I have 9 individuals. Each one has 2 alleles. 13 of them are big W, so 0.72%. Now try and figure it out for the little w allele. So I have 2 little w's, 1, 2, 3, 4, and a fifth one, 5. So 5 out of 18 is 0.28, or 28%. Now I could also, of course, just subtract this from 100, so only 2 alleles in this population. And these numbers are really useful because we can see if the allele frequency is changing. We can tell a population is evolving if their numbers are changing or not. If the number stays the same, or fairly close to the same through multiple generations, there's nothing happening, it's not evolving, there's no real selection pressure, nothing is changing the environment, everything is kind of staying the same, the population is not evolving. But if these are changing, and maybe white becomes more common, then the population is evolving. Okay, so here's another example. Take a minute, calculate the allele frequency in this population, the same diagram you had earlier. Pause the video and calculate the allele frequency of big A and little a. Okay, so for big A, hopefully you found 18 out of 34, which is 0.53, is the allele frequency. And for little a, you found 16 out of 34 is 0.47. It's a pretty even split in this particular population. No one allele is really outnumbering the other like in the purple and white flowers. Okay, here is multiple generations. Look at the allele frequency. So here we have a population of rabbits. In its initial population, you can figure out the allele frequency, and then only these 5 rabbits reproduce. The other 5 do not reproduce. Maybe they get eaten before they can reproduce. Something they don't survive. Something happens to them. So these 5 reproduce, and they produce this generation. Among this generation, only these 2 rabbits reproduce, and they produce this generation. So I want you to calculate the allele frequency for each generation. Pause the video and do the math. Okay, so hopefully you figured out the first generation, my A gene, the big A, is 50%, and the little a is 50%, 0.5 and 0.5. If I count them up, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 big A's out of 20, which I have 10 little a's out of 20. The next generation is going to change though. Now I have 0.7 big A and 0.3 little a's. And the next generation I have 1 big A and 0 little a. So I have 100% big A. Everybody here has the same allele. My variation has decreased dramatically over population. Must be some reason why white rabbits are never breeding. Maybe they're really easy for predators to see. Something like that might be happening to them. Okay, so how are gene pools affected by evolution? What's the connection here? Why are we talking about gene pools in a unit on evolution? Well, if the gene pool is changing, the variation there is changing, population is evolving. And if it's not changing, it's not evolving. And the gene pool is where the variation comes from. Remember, variation were the 4 principles of natural selection. If every individual was the same, no one would better adapt it. So we have to have that variation for this to work. Okay, so the different alleles that exist in a population, they can be advantageous. Maybe they help an organism run faster, bigger leg muscles. They can be disadvantageous. Maybe it's a coloration that makes it stand out in the environment and predators can find it more easily. Or it can be neutral. So this is not really going to affect it, help it, or hinder it. It kind of is a little bit different. So using the rabbits as an example, I want you to kind of talk through natural selection. How is that going to lead to these changes in allele frequency over 3 generations? So take a minute, pause the video, and think about this. How would natural selection cause these changes to happen? Okay, well, we need to have our 4 parts of natural selection. Our first one would be variation. So the population, we have variation. We have white rabbits and brown rabbits. We have some that carry only the brown alleles and some that carry the brown and the white, like the big A, little a, like this guy. We need to have overproduction. There's too many rabbits that can survive. Somebody's going to take out these 5 rabbits here. We need to have adaptation. So something about being brown is going to have to make better adapt to their environment. And we have some modification. So those rabbits who are brown, when they reproduce, they pass on their traits to the next generation. So maybe if we think about it, maybe it's an environment where white makes them stand out a lot more, so they're more likely to be caught by predators. So one generation, only brown ones live long enough to succeed and reproduce. And the next generation, it's even more predators for some reason. They're easier to find. Maybe it's a bad weather year and there's not enough tree cover to hide the rabbits, not enough bushes and grasses. They're easy to catch. Only 2 brown rabbits survive. And then now we have all these ones here. Okay, so frequency can change based on those parameters. Something in the environment, what we will call a selection pressure, is causing them to evolve or adapt. Okay, so that kind of selection can happen on a bigger scale over many generations. So this is the second image to draw here, these types of curves. The red is the original population, and the blue is the after adaptation population. So if we look at allele frequencies, we can better understand which traits are better adapted. So the more common trait is going to be better adapted to that environment. And that kind of pattern analysis of allele frequencies has led to 3 different types of natural selection we kind of classify different examples in. And that would be disruptive selection, stabilizing selection, and directional selection. So if we look at this original kind of red curve here, this is where the mean of the most common traits are the top ones, would be the center. Okay, so in disruptive selection, the mean trait, whatever it let's say it's tail length. This is a really long tail. This is a really short tail. This is a medium tail. The medium tail is the best one in the kind of in this original kind of red curve. The mean, the middle length, is selected is ideal. In disruptive selection, though, what happens is something happens in the population where the 2 extremes are selected for. So maybe the mean is selected against, having a medium length tail is a disadvantage. Maybe it's not long enough to help you climb, but it's long enough person can grab you easier. But if you have a really long tail, you can climb better. If you have a really short tail, you're not going to get caught better. So those become the new population kind of distribution of alleles, where we have lots of long tails and lots of short tails and barely any middle tails. Okay, directional selection, we have 2 extremes. So let's say we have the long tail or the short tail, they're going to be selected for, and the mean is selected against. So let's say in this case, originally having a short tail was the best thing. But then some mutations, the changed environments maybe, and the new environment, there's like vines and trees that these organisms can climb, and having a longer tail became better. So over time, having a short tail became less common, and having a long tail became more common. Whereas originally, having a short tail was very common, and a long tail was very rare. So something is causing the selection pressure to have a longer tail, short tail difference here. And one, maybe it's an environmental change. Okay, so stabilizing selection is where you have the mean is the ideal one. So think about tails still, where short tail and long tail both aren't good, but a medium tail is really good. So the middle trait is going to be the best, and that's going to have a population changing from this kind of general curve to a more concentrated population in the middle. Most individuals will have that particular trait. Now, what's going to cause these particular changes in population in the allele frequencies? Well, it's the environment. The environment changes. It's going to cause a different selection. Maybe it's a new predator is introduced. Maybe climate change happens. Maybe a human impact happens. Maybe the population moves to a new area. Something is different, and it's causing the different traits to be more favorable. Okay, so let's look at some examples here. So here I have original population is the blue line. It's the distribution of the number of eggs laid. So in robins, how many eggs they lay per season. Originally, the middle was pretty common, but it's getting a little bit down as it goes to less eggs or more eggs. And then after natural selection, we get this new shape of the curve. A lot of eggs and a very little bit of eggs are gone completely, and most people are around here. So what kind of selection is this? Well, that is stabilizing selection. Most robins lay 4 eggs. If there's too many eggs, the chicks may be malnourished. They can't get enough food from their parents. If there's too few eggs, there might be no offspring who are strong enough to survive. So it's stabilizing selection. Okay, look at this one. Here we have moths, and they have 2 different colors. We have original population here, which is lighter colored moth, and then after natural selection, now we end up having more darker colored moths. So what type of selection is this going to be? This is what we call directional selection. So originally these light colored moths were very common because they could camouflage really easily against birch trees and light colored trees in a kind of clean environment, a pristine environment. But when we have the Industrial Revolution happening in England, and we get all this soot produced from the chimney stacks of the factories, all the trees started to get this darker color covered by the soot from the fires. So then the dark colored moths were more likely to be able to hide better from predators, and they became more common. So went from this original to this is the new curve. So this is definitely directional selection. One direction is much more favored than the other. Okay, here I have fur color in rabbits. So here we have my original population of rabbits here, and then my after I have these two extremes. I have this one and this one in this kind of shape. So what type of selection is this going to be? Okay, so in this population, we have a gray rabbit and we have a Himalayan rabbit. These are better able to blend in with a rocky environment than the whites. The white is a disadvantage. It's too easy to see, but a gray and Himalayan are harder to see. So this is going to be an example of disruptive selection. We're spreading out. The mean is selected against, and the two extremes are favored. Okay, so we'll just practice a little bit more. There's 3 more examples with images on a worksheet I've uploaded to Schoology. Take a minute into this. It's not being graded, but I want you to practice more with this. So your homework is to upload the notes, upload your practice questions. I'll give you feedback on that before the quiz, and then make sure your notebook is organized for the quiz. Remember, it's open notebook, so you have problems so you need to think, which is why I want you to practice problems today to help you guys practice thinking and analyzing different examples of evolution. Okay, as always, if you have any questions, reach out to your teacher, and good luck and have a good day.